Okay, thank you very much, Fabio. And again, good morning to everyone there. As Fabio mentioned, I'm going to talk a little bit about evolutionary robotics and also biological robots, otherwise known as Xenobots, that you see here. And I'm happy to take questions. I can see the chat, I can see the Zoom chat window. So please feel free to type any questions you have in chat, questions about clarification. I'm happy to go over those, and then I'll leave some time at the end for question and answers. And speaking of time, I will check with Fabio. I think I have 1 hour total. Is that right?
Yeah, yes, yes.
Okay, okay. All right, so I've been working in the space of evolutionary robotics for about 20 years now. I got my start as a PhD student in Professor Rolf Pfeiffer's lab at the University of Zurich. And when I finished my PhD, we co authored a book together called How the Body Shapes the Way We Think, in which we tried to enumerate all the ways we knew at the time that the body can influence the way that organisms think and the way that we might build machines to use their body to exhibit adaptive behavior. And Fabio has already given you a lot of compelling examples of how that's possible. So, what I wanted to do this morning, my morning at least, is to focus on the evolution of my thinking, how evolutionary robotics and xenobots more recently have shaped the way that I think about how the body shapes the way we think. So I wanted to start with state of the art, where we are at the moment. As we all know, robotics has come a long way in the last few years, thanks to the incorporation of modern deep learning into robotics. This is just one example. This is Google's Palm E robot from a few years ago, doing a very good job of taking in verbal prompts and exhibiting appropriate actions.
Sure, sure.
We all know how this works, more or less. It's using deep learning and large language models and found other foundational models to respond appropriately to verbal commands. Given that, even state of the art robotics struggles with what's called out of distribution prompts. So, for example, this is just something I made up, but you could, for example, prompt Google Palm with the command look behind the drawers, and it may or may not have been trained on this, but I doubt that it has. And from the screenshot you can see here, there is no way that Palm is going to be able to reach its hand into this drawer and behind the drawer. And even if it could, its camera is mounted on its head, so it would not be able to see behind the drawer. So the appropriate response to this prompt is, I can't. That being said, Palm could, in theory, look around itself and try and find a tool or something that could reach behind the drawers. But again, how would it possibly know? If you observe a human child, however, you will often see that a human child will receive something that it's never heard before, like look behind the drawers, and exhibit zero-shot learning without any additional learning. It will appropriately respond to a command that the child has never heard before. So there is still a bit of a mystery about how organisms are able to seemingly respond appropriately with little or no training to out of distribution situations. The question arises, are they actually new experiences for the organism or possibly for the robot? And that's where the body comes into play. So we'll switch from robots to organisms for a moment. I want you to put yourself in the shoes of this particular organism. This is a salamander that's developing. You can see that there was originally one cell that split into two, that split into 4 and 8 and 16 and 32 and so on. I want you to now put yourself in the shoes not of the salamander as a whole, but as an individual cell participating in this developmental process. Consider the breadth of sensor motor experiences that individual cell is having during development. That cell is pushing and pulling on its neighbors. It is communicating, as you can see here. Some of these cells are migrating through the body of the developing organism. The neighboring cells, in turn, push and pull on that individual cell. That cell has a huge amount of experiences with doing all sorts of things, including working together with its neighbors to determine whether or not it can move into a small gap in the tissues of the developing organism and quote unquote look behind the drawers, see inside that gap and determine what's going on. So in the case of robotics and even state of the art robots, I would argue that they are, even if they have a body, they are still hollow. Not literally hollow, they're filled with metal and plastics and ceramics and sensors and motors, but intellectually hollow. At the level of the whole body of the robot, if it experiences something that is outside the sensor motor experience that it experienced during training, it's unlikely to know what to do. It will possibly do something unexpected and possibly dangerous. On the other hand, the developing salamander that you just saw, and all of us, every organism that does exist or has ever existed, is not intellectually hollow. At every level of organization inside an organism, every organ, every bit of tissue, every cell, and every subcellular component has experiences a very broad range of sensor motor experiences. So if the real world throws a new experience at the organism as a whole, that overall whole body sensor motor experience gets broken down into smaller and smaller and more local sensor motor experiences as that sensory information propagates inward through the body. And someone at some level of organization inside the body of that organism is going to quote unquote know what to do and communicate a plan back out to the entire organism. So I often refer to this as morphological pre-training. Organisms are born into this world with tons of sensor motor experience already at the sub-body level, and that pre-trains them. All of that internal flux and internal adaptation pre-trains them to handle all of the surprises that the external world throws at them after they're born into this world. So the goal of my lab over the last few years has been to figure out how to morphologically pre-train machines, to give them particular kinds of bodies that are not hollow, in which inside the body of the machine, they can try out adaptive processes or adaptive responses to surprising things and use that to handle surprising external things. We started this journey with the Xenobots because these are biological machines that are biological robots that are built from biological parts. They are built from cells, like you can see here. And you can see in this particular video a Xenobot, a biological robot, exhibiting zero-shot learning, handling what looks like a surprising event that they've never experienced before. What do I mean by that? What you're looking at more specifically is a collection of about 2,000 frog cells. This whole robot is about 1 millimeter in diameter. And halfway through this video, Doug Blackiston, a staff scientist in the Levin lab at Tufts University, is going to come in with a very small pin and puncture the Xenobot. And at the moment of puncture, you're going to see this huge white flash in a moment. And that white flash is is calcium being broadcast through the body of the Xenobot, and somehow these cells collectively, there's the puncture, somehow collectively all of these cells are collectively experiencing a situation they've never experienced before, a pin puncture, but they somehow immediately know what to do, which is to migrate into the wound and start to stitch it up. This Xenobot has never experienced a puncture before, but these cells and the cells before them and the cells before that somehow have experienced local kinds of damages and have survived to this day because they presumably exhibited the appropriate adaptive response. So if we build machines out of biological components like cells, the components of that machine have 3.5 billion years of experience of handling external and internal surprise. Just as a point of reference here, you can see this particular Xenobot, again, about 1 millimeter across, trapped under glass on the left-hand side here, just to give you a sense of the scale of these machines. So I'd like to start with talking about these Xenobots and how they came into being. This was a joint project between my lab here at the University of Vermont, focused on robotics and AI, and Michael Levin's lab at Tufts University. Professor Levin is a biologist and focuses, among other things, on regenerative medicine and bioelectrics. That is, how cells communicate electrically to exhibit adaptive responses, not just neurally, but also throughout the entire body of the organism. We started this project back in about 2018, actually, and at that time when we teamed up on a jointly funded project, we taught the Levin Lab a little bit about what we could do in terms of robotics and AI, and the Levin Lab taught us a little bit about what they could do on the biology side. And at that time, Doug Blackiston showed us that he could extract frog skin cells and frog heart muscle cells from early frog embryo, and he could take those two cell types, skin and heart muscle, and he could surgically, under the microscope, rearrange those small bits of tissue into completely new rearrangements. This is known as ectopic surgery or rearrangement of living materials. So we asked the following question back in 2018, which is, if a human microsurgeon under the microscope can rearrange living tissue and have that rearranged bit of tissue survive for about 7 days, could we task an AI with discovering novel rearrangements of these two types of frog tissue to produce something that isn't a frog, that also survives for about 7 days, and that exhibits some desired behavior? So in the same way that we try and build machines or robots out of motors and sensors and batteries and electronics, we rearrange those non-living components into a machine that does something we'd like it to do, like the Roomba cleaning up your carpets. Could we do the exact same thing, but now with living components? And the answer 6 years later is yes. So I'm going to show you how this started. As I mentioned, our building materials were frog skin cells, which, like human skin cells, are passive and soft and can be pulled and pushed by more active tissues, and frog heart muscle tissue, which spontaneously increases and decreases in volume, which when you put frog heart cells together in normal frog development, it gives you a frog heart, in which all of the cells synchronize and get smaller together and bigger together, which gives the heart its primary function, which is to pump blood in and out. So given that, we created an evolutionary algorithm. I think Fabio already mentioned this. It's a very old technique in AI. It goes back to the 1970s. You basically create a population of random solutions to your given problem. In our case, random solutions are random configurations of frog skin cells, which are simulated here as blue cubes, and simulated frog heart cells, which are simulated as these red-green cubes. And if you look carefully at these videos, you'll see each individual red-green cube gets a little bit bigger. We paint it green when it's getting bigger, and we paint that cube red when it's getting smaller or decreasing in volume. So you can see in these videos the blue simulated frog heart cells or patches of frog the patches of frog skin cells being passively pushed and pulled by the more active simulated frog heart muscle cells. This is the very beginning of the evolutionary algorithm. It's generated 100 random reconfigurations of skin and heart, and I'm showing you just 3 of those 100 random configurations. We've also given this evolutionary algorithm a fitness function or a loss function, which is the way for the evolutionary algorithm to assess the quality of every one of these random solutions. The initial objective function was to find a virtual rearrangement of skin and heart tissue that would cause this simulated machine to move from left to right along the bottom of the simulated petri dish. We wanted to see if the AI could come up with a way of putting together skin and heart to produce a machine, something that would locomote or move. As you can see, and not surprisingly, random configurations do not move at all from left to right. They all do terrible at what we want this machine to do. But if you look very carefully, you'll notice that the creature in the bottom, at the bottom here, is actually moving a little bit from right to left. This is just my poor camera work. If you mentally flip the video at the bottom around horizontally, the one at the bottom is moving a very, very small distance to the right. So it's doing a little bit better than the upper two. So what happens, the next step in the evolutionary algorithm is it deletes the upper and middle robot and makes randomly modified copies of the bottom robot. Those randomly modified copies are again simulated, and if they do better than the original parent, that parent is deleted, and we repeat this process over and over again. Here's a video of this evolutionary process sped up. You're not seeing the simulation of any one of these machines now. You're watching a fast forward through the evolutionary process. At every tenth of a second here, you're seeing sort of the best body plan that the machine could come up with. You'll notice that there are many machines being simulated in parallel. When we run evolutionary algorithms these days, we typically run it on a supercomputer where many different nodes within the supercomputer are running parallel evolutionary experiments. You can think about this as a long chain of islands in an archipelago, and on every island there is a separate evolutionary process at work. Back in 2019, when we ran this particular evolutionary algorithm, after about a week on the supercomputer sent us back this MP4, this video. And as you can see, this evolved machine does indeed do what we want it to do, which is to walk from left to right along the bottom of the petri dish. You'll notice it's got an interesting design. We have some passive skin cells on the top that are passively absorbing and combining all the impulse forces coming from the active tissue, which is on the ventral or bottom half of the Xenobot. I heard Fabio mention passive dynamic walking. That's been an area of interest in robotics for many, many years. We were particularly delighted when we got back this design from the evolutionary algorithm, because the evolutionary algorithm had rediscovered passive locomotion, or at least partially passive locomotion. The passive skin material on the top of the Xenobot is somehow combining the sort of desynchronized actions of the heart tissue on the bottom, and somehow that passive skin material is helping to channel that those active forces into non-random, more or less straight travel from left to right along the bottom of the petri dish. So this was the end of the first half of the experiment. The evolutionary algorithm has generated an in silico design. The second half of the experiment was to see whether we could validate this in actual in vitro or living materials. So Doug Blackiston went to work, our microsurgeon, and we're now looking down through the microscope with Doug at about 1,000 frog skin cells. At this point in the construction process, Doug has liberated or pulled bunches of skin and heart cells out of early frog embryo. Every individual white circle that you see in this video is an individual frog skin cell. Doug has pulled these cells into a very small syringe, and then he's pushed these cells with the syringe into the bottom of the petri dish. So we're looking down onto the petri dish, and this larger black circle that you see, this is a small concavity or a small well, and you can see these cells settling into the well. So Doug is looking at that MP4 that we sent him, this one, and he's trying to build a version of this bot. Xenobot. So he's, I'm sorry, not depositing frog skin cells, but depositing a little bit of frog heart cells on the bottom. Then he's going to layer or deposit another layer of heart, another layer of heart, then a layer of skin, a layer of skin, and he's going to try and build up like a sandwich more or less the distribution of cell types that the evolutionary algorithm predicts will produce motion. This is later in the construction process. Doug, all of these cells, which are kind of sticky, have congealed and pulled together into this about 2,000 cell wide machine. And you can see that Doug is now using some very small microsurgical devices. These are basically, at this point, just very, very small pliers, very small pincers. And like a sculptor, he's scraping away the unwanted material to now get the 3D geometry of the robot. You can sort of see the 4 stubby legs pointing up towards the camera. And when he's finished, he's going to turn this little Xenobot right side up on the bottom of the petri dish, and we're going to see whether it walks like the evolutionary algorithm predicted that it would. And of course, I wouldn't be showing you these videos if it didn't. So here it is. You're now looking in the bottom half of this video at the Xenobot, the in vivo Xenobot that Doug just built, placed right side up in the bottom of the petri dish. And it doesn't look exactly like the evolutionary algorithm described, and it doesn't move exactly like the evolutionary algorithm predicted, but the match is close enough that you get an in vivo biological robot that does what we want it to do. When we published this work back in 2020, there was a lot of public interest, as you can imagine, and a lot of confusion about what this thing actually is. Is it a frog? Is it not a frog? Is it a robot? Is it not a robot? For my part, I argue that it is a robot because it's something that we built, we being the AI plus Doug, the microsurgeon. We built and we designed and built to do a desired task. What makes a robot, in my opinion, is not the materials from which it's built, but what it does, which is to do something on behalf of its human agents, us in this case. So these were the original Xenobots. They're interesting for a lot of reasons, I think. One of them is that it's relatively easy to get them to do other things that you see simple robots like the robot vacuum cleaner Roombas doing. Here you can see we took a couple, some of the evolved simulated Xenobots and put them together, and they exhibit a desired collective behavior, which is cleaning up a messy environment. You can see the in vivo Xenobots doing more or less the same thing in the bottom half. If you look carefully in the bottom half of the video, you'll see there's some very small particulate matter, and the bots are doing a okay job cleaning up. The other thing that's interesting about the Xenobots is they're exhibiting this behavior with no neural structure whatsoever. As I already told you, these bots are made from skin and heart muscle tissue. There is no neural tissue inside these bots. There's no electronics, but we're getting interesting behavior. The Xenobots over the last few years, again, have changed my thinking about how the body shapes the way we think. If we look at nature, we see a lot of interesting and adaptive behavior in organisms that have no brains. And here is yet one more example of such an organism. We moved on from that initial experiment to a second experiment that we reported in Science Robotics back in 2021. This was an attempt to genetically, sorry about that. Let me reshare my screen. Sorry, can everybody see that? Yes, okay. All right, so in the second experiment, we genetically modified the Xenobots to perform a relatively simple robotics task, which is go, see, remember, come back, and tell. So what does that mean? Let's start with Go. Doug Blackiston, our trusty microsurgeon, genetically engineered Xenobots to fluoresce green, as you can see here. You can see them fluorescing green. He genetically engineered them to fluoresce green and then placed them in a petri dish that you see at left. And they went. They started moving around in the dish. These particular Xenobots have been genetically engineered so that if they ever come into contact with blue light, as you can see on the left, they switch from fluorescing green to fluorescing red. And they will continue to fluoresce red even after they've passed out of blue light. So not only do they go and see blue light, but they quote unquote remember. They continue to fluoresce red even after they've left the blue light behind. If we then collect these Xenobots, more or less go back, once we collect them and pull them out of the petri dish, and if we then take that swarm of Xenobots and count the ratio of green to red Xenobots, we have an estimate for how many of them came into contact with something that we're interested in their environment, which is blue light. This was just an attempt to sort of see how this new platform could be used to create, in the not too distant future, useful robots. There's them fluorescing red. So that's all I'm going to say about Xenobots. This is an ongoing research collaboration. We have a number of funded projects together with the Levin Lab to push Xenobots in lots of different directions. Last year we reported on anthrobots. These are biological robots that are made from human tissues. I'm happy to take questions at the end about other directions we're going on with the Xenobots and anthrobots. But I just wanted to share them again, that when we think deeply about the role that the body plays in adaptive and possibly intelligent behavior, it can take us in a lot of different directions from traditional robotics and traditional AI, like, for example, the Xenobots.